Okay, for taxonomic evidence, over the last few decades, the affinities between plant groups have been redefined as more and more information is accumulated from various sources. In this section, in my section especially, I will try to discuss the roles of various features to elucidate taxonomic affinities between different plant groups. So I will give a brief explanation on morphological evidence, anatomic evidence, palynology evidence, micromorphology, and ultrastructure, and finally the chemotaxonomy. I want to discuss on molecular systematics on plant, but the general concept has been discussed in the previous class, so therefore I will not repeat in my section. However, if you need more explanation or specific explanation for plant molecular systematic, please let me know, and I will explain during the discussion session. Okay, uh, first, we talk about morphological evidence. So we already discussed this morphological evidence, especially on various descriptive terminologies for uh, different structure of plants in previous classes. So basically, morphology has been the major criterion for classification over the last many centuries. So even nowadays. So the initial classification were based on gross morphological characters. Although floral morphology has been the major material for classification, other morphological characteristics have also contributed in specific group of plants. So the diversity of morphological features has already been discussed in detail under descriptive terminology, as I mentioned before. So here I will just give a brief explanation on how the morphological characteristics can define the classification of the plants of different groups. So first, talk about habits or in terms of life forms. Though there is very little significance when talk about uh, life forms for the taxonomy. However, it allow a means of estimating adaptiveness and ecological adjustment of the plant to the habitat. In pines, for example, bug characters are used for identification of species. Another one is like a woody and herbaceous characters have been the primary basis of recognition of Lacnosae and Herbaceae series within the dicots family. So for several decades, it was believed uh, that trees or shrubs with simple leaves represented the most primitive condition within the angiosperms. So next we'll talk about underground plants, underground parts. So example on how the underground parts of the plants were used in the affinities of the plants. For instance, the first here will give you the example of the iris here, the rhizome characteristics of the iris. These characteristics are important for identification of various species of the genus. Second is the garlic here. Similarly, the bulb here, the bulb structure is an important taxonomic criterion in the genus Allium. So leaves, of course, is very important for identification. So, for example, in palm, the salix and populus can be differentiated based on their leaf characteristics. The Azadiraceae, this species has been this genus can be separated from Melia for the presence from the other Melia. This is under Melia group based on the by the presence of the unipinnate leaves against bipinnate leaves in the Melia. So similarly, the genus Sorbus has been separated from the Pyrus on the basis of the pinnate leaf. Okay, on the Sorbus. Stipules. Yeah, the characteristics of stipules are important source for identification in Viola and Salix. Leaf venation, also important to identify the species between Ulmus and Tilia. And another the final example that I want to give here is the uh, identification of family Rubiaceae is based on the interpetular stipules. So this is useful for identification of Rubiaceae family. So these are some of the example among others how to use leaves as a taxonomic evidence. Next is floral characters. Of course, these are extensively used in delimitation of the taxa. This may include the use of calyx to differentiate or to identify or to characterize Lamiaceae, for the corolla in the Fabaceae, especially in Corydalis. When talking about stamens, also can differentiate or characterize Lamiaceae, Fabaceae, Mimosidae, and also the carpels. Okay, for the Cryophyllaceae. So another characters that you can look at is the in terms of genobasic style, also can be characterized in Lamiaceae. So different species of Euphorbia have a distinctive Cytium inflorescence with clusters of male flowers, each represented by a single stamen. And finally, the fruits. Fruit characteristics are very widely used in identification. In Astraceae, for example, the shape of Crypsela, usually called the akin. So the shape of akin, presence or absence of pappus, and whether the pappus is represented by hairs, scales, or bristles. The presence of absence of beak and its length, and the number of ribs of on the Crypsela, okay, constitute valuable identifying features for the Astraceae. So the number of capsule valves also is used in segregating genera in family Cryophyllaceae, like in the Melodrium, Silene, and Crestium. So and also in the seed of the fruits, also the characteristics of this seed of fruits also is valuable in identification, especially in genus Veronica. So we have a lot of other characteristics of fruits. You can you can refer to the previous lectures on descriptive terminology. Again, like I said before, morphological characteristics are the major criterion for plant classification and taxonomy. I will not intend to discuss details on each of it because I already discussed it in the descriptive terminologies. You can refer our previous lectures for the specific details on how the characters being used for taxonomic classification.